So I am Nam Shiva. I am director of Center for Biomedical Informatics, Vingroup Big Data Institute. So my title talk today is population-specific risk prediction models experiences from the 1000 Vietnamese genome project. So, the risk prediction models provide a quick and simple way to evaluate patients' risk. So, for a disease or event that can guide treatment. So it becomes an important part of clinical decision making. So, Damen AO found around more than 300 models for predicting a patient's risk of developing cardiovascular disease. And also Gostenial found more than 100 models from 2009 and 2014 that use routinely collected data from EHR. So currently, the risk prediction models could be accessed using population-level statistics. So the validity and usefulness need to be evaluated, and calibration and discrimination need to be measured. So here's an example of the risk prediction model. So we call it genetic risk score, GRS. So here's an illustration of the GRS. So we can see that we can take the candidate GPS, and then we do validation. And we can see the two distribution of the control and case, so that we can see the low risk in the right side, the left side, the intermediate risk, and the high risk in the right side, and the best GPS should be in the middle. So also, and then we can use the polygenic risk to do the clinical risk as a clinical risk factor. So you can use it to estimate the low risk or high risk of any patient. So one problem here, very big problem, is a population bias in risk prediction models. So most training data now are European. So disease risk prediction accuracy in other populations is not clear, and usage of training data is typically one way. So European or target population, just small sample size. And prediction accuracy reduced from European to the others. So due to different causal effects or patterns of the LD and so on. So one important thing is model parameters are transferable. So some factors that as related to it is LD or allele frequency or genetic architecture selection or gene environment interaction. So here's a very big problem right now is a European bias in genetic studies. So we can see in the figure, so the left side is a number of individuals in the US in the number of billions. So and the right side is a global population. So we can see now European the number of the population is similar to African and less than East Asian or South Asian. But the most number of individual in the US study is from European. So this trend doesn't change through time. So now European individual dominate all as a population. So we can see that because of that, some model should be changed. And also some observation we can see here. For example, in GIS for type 2 diabetes here, the mixed population model versus single population model. So here we can see that European and Latino. So the adjusted R square that for European and Latino is better than European. And also you can see that if we consider PIS score as a mixed with alpha 1 and alpha 2, alpha 1 is for PIS of Latino and alpha 2 is for principal component, just related to the ancestry, the proper ancestry. So we can see that the GIS here is better for the mixed population. The same observation can be seen here for also type 2 diabetes. We can see that trans ancestry is much better than ancestry specific accuracy. You can see here the trans ancestry GIS in the right side and the bottom. It's much better than other like African ancestry or East Asian or European or Hispanic, Latino or South Asian ancestry. And we can see that the mixed population or the trans ancestry level of the GIS will perform better than the ancestry specific. And here that's the reason why we are doing now the 1000 Vietnamese Genomes project. So this project already completed in December 2021. So we have whole genome sequencing of the more than 1000 Vietnamese with 1000 terabyte data from across North and Central and South. And just is relative to the population in Vietnam. So here's access can be accessed through all other population. So here's a typical results you can get here. So it's a high rate of the novel common variants. So we may need to revise genomic medicine practice in Vietnam because we can see here, even for very common variant, very common SNP or indel. So there are even 15% of the novel variant here in Vietnamese compared to 1000 genome project of the initial 1000 genome project data. And even for common, it's 22% of it's not just novel. That's why we have to consider population specific model and data. And here is our project of disease risk prediction for Vietnamese that we are sequencing and genotyping more than 2,000 samples. And we consider Vietnamese specific disease-associated variants with common disease like type 2 diabetes, cancer, and cardiovascular, Alzheimer, Parkinson. And we are developing SNP chips and PIS model for disease risk screening and using the trans-ethnic genome-wide analysis. So here's a project that we collaborate with a leading university, medical university in Vietnam, is the Hanoi Medical University, and the University of Queensland in Australia. And here is a typical example that we are doing now for the disease risk prediction. And here is a model that we have SNP chips and disease prediction. And we can see here, we can use 1000 Vietnamese genome project data and disease-associated variants from some public database like DB-SNP or 1000 genome project or some other. And also our partner clinical data from some leading hospital in Vietnam, some also from some leading medical university in Vietnam. And we are developing a SNP chip named Vingroup chip, and also a predictive models for disease risk prediction. And here we aim to get some model and some chips that are affordable for genotyping millions of people, and also have the higher accuracy for Vietnamese. And our result here shows that VGC is super sensitive for Vietnamese. And one conclusion here is a genome-wide study for Vietnamese may need to consider VGC instead of some popular commercial SNP chips. Here is a result. We can see that the pink thing here, just Vingroup chip is better than the other from AIxImpact or from Infinium, GSA, from Illumina or Informatics, and across all allele frequency, major allele frequency, minor allele frequency bin. So here is a interesting result. Also, we can see that based on this information, we can get some take-home messages here that the GIS could be used responsibly to improve human health. Some factor we should consider is uncertainties and biases, and also misunderstanding and misuse of the information from GIS. The second thing is the GIS accuracy gaps between European versus non-European sample are available there. And we should combine training data from European and the target population to help improve the GIS model. And also improve risk prediction model in general. And population-specific risk prediction models should be considered in this way. So also another thing is the trans-ancestry models could improve the transferability. For example, it can lead to more effective clinical translation, like some examples that already shown for type 2 diabetes or cardiovascular disease. And we also can help improve systematic assessment of candidate causal genes. So here is a very important for trans-ancestry model because, as I mentioned, the European data now dominate all others. So it takes time, even 10 years, 5 years to 10 years to reduce the domination. So now the only thing we can do is doing the trans ancestry model, and we combine training data from European and the target population to improve the models. Okay, so here is our network of Big Data and some other leading institution and as a leading collaborator to realize genomic medicine in Vietnam. As you can see here, we can leverage the 1000 Vietnamese genome project and the genome assembly project that we are doing here. Also the patient sample and chip testing healthcare from our collaborator with some hospital in Vietnam, and the model optimization and genomic analysis to improve and to promote the genomic medicine in Vietnam through some clinical testing, some patents that we are applying through our analysis and publication and international network. So here's our leading institution in our network from US, Europe, and the Asia and the Vietnam, and all of them here. We are working together to promote and to realize genomic medicine in Vietnam. So that's all information that I present today. Thank you for the listening.
Thank you very much, Dr. Võ, for your presentation and for sharing with us today. I can see that there are a few questions from the audience that we'd love to hear your thoughts on.
Thank you.
And I think we have time for 2 questions. The first one is, what do you see the challenges of genomic medicine in the future?
Okay, that's a very broad question, so difficult question. But I think I can see several challenges. The first is, like I mentioned in my presentation, the data. So now we see that the most data is from European samples, not from other, even from South Asian, East Asian, and Southeast Asian, and African. We don't have much data, and to implement the genomic medicine, we need to understand about genetics of the ancestry level, like even Vietnamese population. So if we don't have the data, if we don't have genetic medicine projects, we cannot understand more about the genetic structure of Vietnamese or other population. We cannot understand how some treatment or some diagnostic in European can be applied to Vietnamese, can be applied to other Asian population in a safe way, or in some way that we can get the reliable result. So we cannot do that. So the first thing is the data. If we cannot have the data, that enough data to do that, so genomic medicine cannot do. The second thing is even we can have some sample from some other projects, genetic projects, but we can, if we don't have genetic data, for example DNA, RNA data, we cannot do something that meaningful for genomic medicine, because for genomic medicine we need to build some model based on genetic data, and maybe with some other data like phenotype data or some environment data or something, but at least we need genetic data. If we have sample, we have some study, but we don't have genetic data, it's very difficult. And the third thing is the third challenge here, challenging things is here, because the clinical doctor, they may be kind of conservative. It's not in Vietnam or not in Asian country, but even in some developed country. I have been in the US for a long time, and I see that okay, even clinical doctor in the US, they are also very conservative as well. So to implement genomic medicine in Vietnam and even in other country, we need to talk more with the clinical doctor, and for them to see that okay, some benefit of genomic medicine. Thank you.
Yeah, thank you very much, Dr. Võ. So yeah, for the second question, I think it's partly answered by what you have shared before, but I want to hear your thoughts on that. So is the same method you recently shared can be used for other populations?
Yeah, sure. I think as a population, maybe genetic structure between, there's some differences between genetic structure of Vietnamese population or some other population, and there's some big gap between genetic structure European sample and Vietnamese sample and as an ancestry sample. I see some paper before that show very big gap between some population, but the underlying model should be similar. So if we can do some, let's say some transfer learning model between European and some other population, we can do that for Vietnamese or for some other population. So it should be feasible, I think so. Of course, there are some differences between even if we do transfer learning from European data to some other population. It depends on how closely between European sample and the other, population-specific sample. But at least the underlying models and the method are kind of similar and can be applied to many populations.
Thank you very much. Yes, and yeah, thank you. So for now, I would like to invite our next speaker, Freddie Yu, from Johnson and Johnson. Yeah.
Okay. Thank you very much.